Good afternoon. Welcome to this afternoon session. My name is Jeremy O'Friel. I'm the chair of the EEB department. First of all, I'd like to welcome all our visitors, both speakers and members of the audience. Thank you so much for coming here. This is our 13th annual Area Career Scientist Symposium. Every year we have a new topic, and we're very gratified to have so many people come and join us. So one thing I'd like to emphasize is science is a social phenomenon. Okay, a very important part of scientific activity is networking and meeting each other. All our speakers, etc., and the host department are very friendly. Take advantage of this opportunity as we go into the day. Please get to know each other and ask questions and establish connections. I want to thank in particular our organizing committee, and they've done a fantastic job. Every September, October, we have a question, what's going to be our theme, our topic for the following year? And we need a topic area and we need volunteers. And the committee has responded magnificently and they've done a great job. So can we please give them a round of applause? Thank you. And there's one other person I should thank. The brochures and the posters are of very high quality. We have a superb scientific illustrator in the Museum of Zoology, John Megan. He's not here, but he's done a fantastic job. Thanks, John. Now, Andrea, in her introductory remarks, mentioned our long-term benefactor. I'd like to touch on this very briefly again. This is Dr. Nancy William Walls. She passed away, unfortunately, a few years ago. She was very much a pioneering scientist, and she and our longtime former chair, Deborah Goldberg, established a strong bond, and Nancy initiated and supported the first 11 of our Area Career Scientist Symposiums. And without her, we wouldn't have established this tradition. It's a highlight of our academic year. It allows us to network and to interact with, especially with our regional university, college, and departmental neighbors, as well as bringing outstanding young scientists and keynote speakers. Some of those speakers have come on to join us. We have two faculty members who we initially met as speakers in our Area Career Scientist Symposium. And who knows, maybe some of you speaking today may be joining us at some future time. Okay. One of the things I'd like to emphasize very briefly is that the theme this year is on phenotypic plasticity. This theme is not just of academic interest to us, it applies to our department. We're undergoing two once-in-a-century changes in our structural environment, and I'd like to tell you about them very, very briefly. One of them concerns the relocation of our world-class museum programs. We have a Museum of Zoology and an herbarium, and the Museum of Zoology in particular is just about finishing its massive move with millions of specimens to our new research museums center a few miles south of Central Campus. In this center, we will co-house Museum of Zoology, herbarium, Museum of Paleontology, and the Museum of Anthropological Archaeology. And in approximately a year or so, the move will be finished, and we'll have a remarkable combination of research museums with millions of specimens covering biological diversity and human cultural diversity. Okay. The other change that's happening is our entire department is relocating next year into a brand new biological sciences building. It's being built just south of here. Many of you may have walked by it. At the moment, it's pretty cold out there, okay, but you can imagine this in the summertime. There are three main towers. We're moving in there with our sister biology department and the Museum of Paleontology. One of the interesting details about it, it will also include, this is a view from the street, it will also include a public natural history museum, okay? And the structure, this dome here is a planetarium. The public natural history museum will be present on these floors and will interact with our biology departments. So it's not just a science building, it's a building for education and outreach. And in approximately a year and a half, we'll be settled in there, okay? The one last thing I'd like to mention is we're also now, at present, engaging in a major wave of faculty recruitment. We're looking at potentially up to 6 new colleagues, which is very exciting, 3 of them in ecology. And so if you were to come back and visit us, you can imagine that in 2 or 3 years' time, we'll be in a new home. Our museums will be in a new home, and we'll be a significantly different department once everything settles down. So this will be a test, okay? Is how does the genotype change in response to a new environment or habitat? We've got that. We're also changing our academic genotype. We've got new talented people joining us. So come back, please. Visit us again, okay? So let's hand it over to Snell.
Do you have it?
Sure,
There we go. I just need to get the.
It is my pleasure to introduce Ben Parker, who is currently a postdoctoral scholar at University of Rochester. His research focuses on the genetic mechanisms that underlie phenotypic variation, and he seeks to understand how that variation is maintained in natural populations. He's particularly interested in the traits underlying associations between animals and microbes using the pea aphid and other insect systems. When not in the lab, Ben has spent time developing new courses, including classes on microbes and disease and evolution and human health. Today, Ben will be telling us about a very new project he has recently started that investigates the genetic variation in the pea aphid wing polyphenism.
Okay, well, I'll talk about it. So, afternoon session of the Symposium on Phenotypic Plasticity. It's probably not controversial to say that. Okay. Awesome, thanks. So it's not controversial to say that organisms often respond to variable or changing environments by altering their development to produce adaptive phenotypes. Like a lot of traits, there's genetic variation in phenotypically plastic traits, and we had some great introduction to the idea of genotype by environment interactions earlier. In our cartoon here, these different lines represent different genotypes that are responding differently to an environmental cue in terms of the phenotypes that they're producing. And like a lot of variation, this is subject to natural selection, and we think that this natural selection on plasticity is really important for determining how a plastic response can be adapted to kind of a fitness optimum. And so there's a lot of interesting questions to ask about genotype by environment interactions. Maybe in particular, what kinds of genetic mechanisms underlie G by E traits? What kind of genes are the targets of selection? And then if we have lots of variation in these traits in natural populations, what's maintaining this genetic variation? So we're going to look at the ecology of one of these traits. So we talked a lot this morning about phenotypic plasticity that was continuous, so continuous traits. I want to focus your attention now on kind of a special case of phenotypic plasticity we refer to as polyphenism, where discrete morphs are produced in response to the environment. So some really interesting examples come from lepidopterans. These are 3 different species of butterflies, and they have a wet and a dry morph. They look very different in terms of their coloration. My phone is doing something weird. We even saw this exact same picture of Daphnia in an earlier talk that in response to signals of predation produces kind of defensive hood plastically. Social insects, a great example. We're going to hear about beetles in the next talk. And so I think polyphenisms are really fascinating for a couple of reasons, but they also make a really great study system because it's a very clear plastic phenotype that we can study in the lab. And kind of the hallmark of polyphenisms is that these represent a set of discrete morphs that vary in a number of ways: physiological, morphological, behavioral, life history traits. And so the polyphenism that I study is the pea aphid, Acithrocypin pisum. If you do any gardening, you might find pea aphids in your garden. They're sometimes referred to as plant lice. And these two adult aphids are genetically identical. The female on the left doesn't have wings, very sedentary behavior. They don't have a lot of sensory organs on their antennae. This female to the right, which was produced by the same mother just a few minutes after, has wings. They disperse to new plants. They have really active behavior. They have lots of sensory organs. And so this is what we refer to as a transgenerational wing polyphenism. So the signal to produce winged offspring is picked up by the mother. So aphids reproduce asexually in the summer through parthenogenesis. So they're producing genetically identical offspring, really useful for the experiments that we're going to do because we can capture genotypes in the lab for a long period of time, years and years and years. They also go through a round of sexual reproduction in the fall. But all throughout the summer, they go through 10 to 15 generations of this parthenogenesis. And so an aphid by itself will produce primarily unwinged offspring, but if a mother picks up signals of crowding or of deteriorating conditions, she will induce some of her offspring to eventually become winged. There's some really entertaining papers from the 60s where they took aphids and poked them with a paintbrush over and over and over to induce the wing response. And so these winged aphids then disperse to new habitats. And that's great if you're trying to get away from a plant that's about to crash. But they have reduced fecundity. So winged aphids suffer about a 30% decrease in terms of lifetime offspring production. Aphids have about 100 offspring, so that's kind of a large fitness cost. And so today I am going to use the system to tell you about two different stories. First, we're going to ask, how much variation is there in a natural population in terms of this plastic wing response, and what genes underlie this variation. And then we're going to talk about maybe, if we do find a lot of variation, which, spoiler alert, we do, what ecological mechanisms might be maintaining this variation. So there will be, I don't know how satisfying that answer will be. The answer is that it's complicated. Okay, so just to give you some methods about our kind of standard laboratory crowding assay. So we take aphids from all of these aphids in this study will be from one genotype, and you rear them at low densities on a plant. You put them by themselves into a petri dish, and they have primarily unwinged offspring. When we do the same thing, but we crowd them in a petri dish for 24 hours, and then we put them on a plant and count their offspring, they have a large percentage of winged offspring. So crowding them in this petri dish is the methodology that I will use over and over and over to induce this winged response. And one of the great things about the system is that previous work in the lab has identified the developmental mechanisms underlying this plasticity. So how do we go from an environmental cue of crowding to winged offspring? We understand those pathways to some extent. So signals of crowding are picked up by the brain. We think there is some induction of signaling from biological amines like dopamine, neurotransmitter signaling. So in this study, they used mass spec to look at dopamine titers in aphids that were alone in a dish versus those that were crowded in a dish overnight. Dopamine titers dropped in these crowded aphids. A lot of the pathway is also supported by transcriptome, so gene expression evidence that kind of implicates changes in gene expression in response to crowding in genes in this pathway. We also think that ecdysone signaling, or molt hormone, that's common amongst invertebrates, insects, is involved in this signaling pathway. So in this experiment, an ecdysone analog was injected into aphids versus a control, and we see a decrease. We see a decrease in the induction of winged offspring when we inject them with ecdysone. And then the signaling pathway moves into the ovaries, where kind of chromatin remodeling and transcriptional changes happen in the offspring themselves to kind of grow the muscles and the flight muscles and the wings that are kind of important to being a winged aphid. So if you work on aphids for a really long time and you spend hours every week changing them, you kind of get familiar with the personalities of the genotypes that you keep in the lab. And one thing you notice if you're trying to do experiments on winged aphids is that some genotypes are really hard to induce it's really hard to induce winged offspring from some genotypes. So you can neglect them, you can go on vacation and come back in 3 weeks, and the cage will be overrun by aphids, as crowded as it could possibly be, and you don't get a lot of winged offspring. Other aphids, you can make the most pristine, amazing aphid habitat, utopia of being alone in a cage, and they still tend to produce some winged offspring. So they're very sensitive to crowding. There's a lot of variation between our lines. And we wanted to know what genes are involved in these genetic differences between lines that we refer to as low-inducing and lines that we refer to as high-inducing. So the first possibility is that genetic differences in these genes that are in this developmental pathway that lead to the production of winged offspring vary and underlie the differences between genotypes in wing plasticity. So we might find genetic variation in some of these biological amine, neurotransmitter signaling, maybe in ecdysons, something like that. The other possibility is that maybe the genetic variation is in some other mechanism. So maybe heat shock proteins kind of temper the way that aphids respond to stress, and then that underlies the differences in sensitivity to crowding. And so basically, we're going to use the system to ask, where's the genetic variation in phenotypic plasticity? What genes underlie this difference? So we started working with a group, with Angela Douglas's group at Cornell. They were developing a genome-wide association panel for P aphids. And this panel involves 200 genotypes that were collected from a much larger set of aphids that were found to be genetically distinct with microsatellite sequencing, so 200 distinct genotypes. They were all collected from a single field of alfalfa in Ithaca, New York at the same time. It's important to point out that these aphids were collected really early in the spring, right after hatching, so they're not aphids that flew into the field, they're aphids that overwintered there. And so we took this panel, we drove it to Rochester from Ithaca, and we reared them for a lot of generations at low density. And then for all 200 lines, we induced crowding by putting them in these dishes. We left them on plants for 24 hours, and then we took the adults off and counted ultimately whether their offspring became winged or wingless. Okay, so this graph shows all of the 200 aphid genotypes along the x-axis. On the y-axis, we're looking at the percentage of winged offspring that were produced in response to this crowding. And the bars all represent kind of the average over multiple replicates of this experiment, M. And we found a really astounding amount of variation in this wing response among our 200 lines. These are lines all collected from the same field right next to each other on adjacent plants. And they have an astounding amount of genetic variation in terms of how sensitive they are to crowding. We have lines that don't seem to produce very many winged offspring and lines that produce nearly entirely winged sets of offspring. So the point of using this GWAS panel is that we have some genetic information about what's driving these differences. Our collaborators took a restriction enzyme associated DNA sequencing approach, so RADseq, which is kind of a reduced representation of the genomes of these lines. We identified 83,000 SNPs where the minor allele is at least 10%, so a genetic map of our genome. We had roughly one marker every 7 kb across the genome, given the size of our genome. And I'll just point out that linkage disequilibrium is really small. We think it's really small in P aphids. My Drosophila genetics friends, they like to complain about how small LD is in Drosophila. It's even smaller in the P aphid. And this means that we're going to miss a lot of genes, but also that our markers are probably tightly linked to the gene that is associated with the variation. Okay, so in this graph, we're looking at the genomic architecture of this genetic variation in phenotypic plasticity. Our 83,000 SNPs are along the x-axis. Our genome is still broken up into a lot of thousands and thousands of scaffolds, so the scaffolds are in order along the bottom. And basically, each point is just a single SNP. We're just simply doing a correlation between the alleles at a particular SNP and our trait. A more complicated way of saying that is that we used a mixed effects approach, and we incorporated the population substructure in our panel, and we also included the endosymbiont background of each of the lines, which has a really interesting effect on the plasticity that I'm going to completely ignore, but just know that we've compensated for it here in this analysis. And if you're interested in how like the vertically transmitted endosymbionts of aphids play a role in plasticity, we can talk about that later. And so on the y-axis, we're looking at a log of the p-value. How strong is the correlation between the alleles at each SNP, and our plastic trait. And so you can see that there are a lot of genes involved in this variation. And so we can use, oh, and sorry, the lines on this graph just represent two different thresholds for statistical significance. The dotted line is often used in the literature, and the solid line represents a kind of a more Bonferroni-corrected p-value. And I've just highlighted the scaffolds where any SNPs were statistically significant in purple. And so we can use what we refer to in grant applications, at least, as a well-annotated genome to ask what genes are linked with these SNPs. And we found two main categories of genes. The first were transcription-related, so a lot of genes that we think might be involved in modifying gene expression. I'll talk about gene expression in a minute. But really excitingly, we found this group of neurotransmitters that seem to vary. And so in particular, we found that the dopamine receptor, there's only two of them in the aphid genome, we found that a SNP tightly linked, or inside, I think, of the dopamine receptor, this dopamine receptor gene was associated with variation in plasticity. And so when I complain about our uncharacterized genes, our genome is actually not very well annotated, so there are a lot of genes that we don't know what they do. There are a lot of genes that we haven't quite come up with a story for why they are involved, and so clearly we have a lot of work left to validate some of these mechanisms. And to do that, we're taking a pool-seq approach, so we're sequencing pooled DNA from aphids on both ends of the spectrum, so aphids that don't produce a lot of winged offspring, those that do. And that will give us a sense of where the genetic variation in these genes are. Is it in coding regions? We're also doing pharmacological manipulations, so taking aphids that represent both ends of the spectrum, inject them with dopamine and some of the other genes that we're interested in, and see whether they have a different response in aphids that are from different genotypic backgrounds. We're also really interested in gene expression. So ultimately, we hope to do a really broad-scale transcriptome experiment. For now, I'll just tell you about some work we've done looking at gene expression in two aphid lines, one from the low and one from the high end of our spectrum. And so we've taken aphids. We have a solitary treatment and a crowded treatment. And this is a heat map showing fold change differences in response to crowding, with the darker blue boxes representing an increase in gene expression in response to crowding, the red showing a decrease. And so we have these two different genotypes, one of which is a high-inducing, one of which is a low-inducing line. And basically, we found that these genes are more strongly upregulated in our high-inducing line, and they don't show as strong of a change, if any, in our low-inducing line. So more evidence that these genes might be important in this genetic variation. And so ultimately, we hope to do, or we are about to do, a transcriptome using pooled RNA from kind of aphids from the low and the high ends of the spectrum, and that'll give us a sense of kind of which genes are being differentially expressed in response to crowding in aphids with very different phenotypes. So some more work to be done, but I think the story that we are starting to tell is that some of the genes in these developmental pathways may underlie the genetic variation between lines, so that the genes underlying variation in wing induction include some that control the developmental expression of this polyphenism. And we think that that is kind of a fundamentally different, that tells us something fundamental about how evolution is acting on this trait, that the genes tend to be in these same pathways that kind of underlie the production of this trait from a developmental perspective. Okay, so I'm going to change gears a little bit now and talk more ecological work. And so we have all of this genetic variation in wing induction. What's maintaining this in natural populations? And the reason I kind of got interested in the immune system is that the immune response itself is plastic. So we think of the immune system in vertebrates as being all about antibodies, but in insects and other invertebrates have this innate response, which is conserved across animals. And this innate immune response is phenotypically plastic. So a lot of really interesting work has been done on this in locusts. So locusts have this polyphenism related to density, similar to ours. In low-density situations, they produce a green morph, which is referred to as the solitary morph. When locusts get crowded, they produce this high-density or swarming morph. This is like the Old Testament, destroy Pharaoh's crops locusts. And these high-density morphs have been found to be really resistant to a fungal pathogen. in anticipation of high density conditions, they invest a lot of energy into a costly immune response because high density is associated with an increased risk of disease transmission. And so we were interested in kind of using our similar model, but with perhaps more genetic resources, to look at the same question. And our hypothesis was that winged aphids would invest more in immunity in anticipation of their high density conditions. They're produced in response to high density. They experience high density conditions. We thought that they would be prophylactically investing more in an immune response. But alternatively, we know that wing production trades off with fecundity, and winged aphids are investing all of this energy into the production of this flight muscle and of this wing. And it's possible that wing production might make winged aphids energetically limited such that they were not able to invest as much in an immune response. And we can easily test between these hypotheses by asking whether aphids are more or less winged aphids are more or less resistant to disease. So we used a natural fungal pathogen of aphids. In every talk, I tried to tell this anecdote about kind of a sophist back-of-the-envelope calculation that if you took a single aphid, you allowed her to reproduce unchecked over the course of a year. Aphids are born pregnant. They reach reproductive maturity in 9 days, and they produce 100 babies in their lifetime. And if you let that happen unchecked, we would coat the earth in 100 miles deep of aphids in a single year. And things are not great in the world right now, but at least we're not under 100 miles of aphids. And so clearly something is regulating these populations. This is one of the pathogens that exerts a top-down influence on the population dynamics of this insect. And so aphids come into contact with these infectious spores. The spores burrow through the cuticle. They start kind of multiplying in the hemolymph. And if the infection is successful, it turns the aphid into this kind of fuzzy, toasted spore marshmallow that pushes spores out from the surface, and the spores go on to infect more aphids. And so we brought this pathogen into the lab. We rear aphids, winged and wingless aphids, in the lab, and we infect them, expose them to these spores in these chambers where the spores sprinkle down onto them. We keep them at high humidity for a day, and then that allows the spores to kind of penetrate the cuticle. And then we count how many of them produce this infectious spore cadaver that indicates that the infection was successful. So we're comparing the resistance of winged and wingless aphids that are genetically identical. They only differ in whether or not they have this wing. And so on the y-axis here, we're looking at the percent survival, how many aphids survived the infection. The light bars are control aphids that were not exposed to spores. The dark bars are aphids, unwinged and winged aphids that were exposed to these spores. And we see that contrary to our expectation, winged aphids are more susceptible to infection. Unwinged aphids are more resistant to infection. We measured this in terms of survival, also in terms of the percent that produced the sporulating cadaver, more spores among winged aphids. We use qPCR to measure the pathogen load of aphids, how many copies of this fungal 18S gene can we find in an aphid. The pathogen loads are higher in winged aphids compared to unwinged aphids. It's a log scale, so it's quite a big difference. And altogether, this suggests that winged aphids are more susceptible to pathogen infection than unwinged aphids. We were interested in the immune response of winged and wingless aphids. And so we know that in other invertebrates, a group of cells called granulocytes is really important for phagocytosis of invading particles and of expressing the kind of effector molecules that fight off infection. We know that aphids have these granulocytes, and these darker blobs are the spores in the hemolymph. And so here we are counting the relative concentration of these cells in wingless and winged aphids in response to fungal infection. So in unwinged aphids, we didn't find any change in the concentration of these cells. In winged aphids, we find a strong depletion of cells in response to fungal infection. We then did transcriptomics to try to figure out what genes are being expressed in response to fungal pathogens. So we're looking here at unwinged aphids, and they express a lot of genes, and some of them are kind of they make sense as immune genes. And we used qPCR to look at the gene expression of some of these canonical immune genes like phenyloxidase and lysozyme in winged and wingless aphids. The solid bars are wingless aphids. The dotted lines are winged aphids. These genes are being upregulated much more strongly in wingless than winged aphids. So wingless aphids are more resistant, probably because they're mounting a stronger immune response. That was the summary slide. So a really important part of ecological immunology is the idea that immune responses are costly to hosts. So mounting this immune response takes a lot of energy. The immune response itself has an autoimmune effect, so it can cause damage to the host. And so mounting an immune response is costly, and so we expected that because unwinged aphids seem to be mounting this higher immune response, that we'd be able to measure a stronger cost. So we took our fungal pathogen, we kind of mushed it up in some liquid, and we stuck it in the autoclave. And here we're trying to measure fecundity effects of being exposed to the signal of this pathogen, but we have no effect of pathogen virulence. So are there any energetic effects of mounting an immune response to this pathogen that are separate from pathogen virulence? And so on the y-axis here, we're looking at lifetime fecundity, total number of offspring produced by an aphid. We have a sterile stab control and our heat-killed fungal pathogen stab. Unwinged aphids are on the left, winged aphids on the right, and the boxes represent 95% confidence intervals. And we don't really see any fitness effect whatsoever in unwinged aphids, even though they're mounting this really strong response. But we see a really strong cost of immunity in winged aphids. And so basically every hypothesis that we put forth in this experiment came out exactly the opposite of what we expected, but the idea is that winged aphids are paying a higher fitness cost for what is ultimately a weaker and less effective response than their genetically identical unwinged sisters. So I tell you about this because I ultimately want to link this to variation in wing production in the wild, and to do that I need to tell you a little bit more about the pea aphid. We talk about pea aphids as a single species, but aphids are actually a group of genetically differentiated populations that are associated and adapted to live on different species of legumes. So about 10,000 years ago, we started growing lots of legumes, and pea aphids then radiated onto these plants. And we can form hybrids between them relatively well, but there is specialization. So aphids off of clover, for example, don't do very well on medicago or alfalfa. And so we like to use this system as kind of a natural laboratory for looking at evolution. And so the first thing we found is that we made kind of a panel of genotypes from all of these different biotypes, these different host plant-associated populations. Fortunately, they all grow in the same plant, so in all of these graphs, there's no effective host plant because they are all happy to live on one single kind of universal plant. And we found an enormous amount of variation. So the experiments that I showed you before were all aphids from this medicago biotype, and we found this whole range of variation in medicago. But in other biotypes, we don't find very much variation at all. There are biotypes that don't seem to produce much of a winged response. And we have no idea why this is. We have looked for differences in body size, in growth rate, in development rate. We did lots of very time-consuming experiments on host plant chemistry to see if we could get the plant to have an induction effect on the host, and we didn't come up with anything. One thing we do know is that the biotypes are really different in terms of how well they resist this fungal pathogen. So again, we took our biotype panel, the same lines as in this experiment. We exposed them to spores and high humidity. We counted, we assayed how resistant they were to infection. And we find a lot of variation between these biotypes in terms of how resistant they are to fungal pathogens. So I should emphasize here that in this experiment, we're using only unwinged aphids. We're just measuring the intrinsic genetic basis, or the genetic effects on susceptibility to fungus. And so the next slide I will show you is entirely speculative, and I'm just using you as guinea pigs to see if this idea holds any water. But the best explanation that I have, so I'll just tell you that on the y-axis, we're looking at these biotypes in terms of the percent of winged offspring they're producing. The x-axis, we're looking at sporulation, so how resistant they are. And we find this correlation between kind of plasticity, wing plasticity and disease resistance. So biotypes that tend to produce a lot of winged offspring also tend to be very resistant to infection. And it's entirely possible that there might be something that we're not measuring that is making, this is a correlation, not causation, and it's entirely possible that there's something that we're not measuring that might cause biotypes, these different populations, to experience conditions that drive both disease resistance and wing plasticity in the same direction. But the other possibility is that we know that growing these wings makes you really susceptible to fungal pathogen infection. Maybe you need to evolve to be more resistant to disease to kind of compensate for the time that you spend as a winged morph. So I'm sure it will be interesting to hear whether anybody buys this argument, but that's where we're at. And so to bring the talk together, basically, we are using this system to kind of explore variation in wing plasticity from both an evolutionary genetic and an ecological perspective. And the idea will hopefully to bring these two things together to get a better sense of how wing plasticity is evolving across these different biotypes and how it might facilitate adaptive evolution or hinder it. So I just want to thank my advisor at Rochester, Jen Brisson, and our collaborators at Cornell who are working with us on this GWAS panel. The biotype panel is stuff that I did with Charles Godfrey at Oxford, and the immunity stuff is from my time at Emory as a graduate student in Nicole Gerardo's lab. So thanks. 2 minutes.
And really cool stuff. Thinking about the difference in inducibility of wingedness across different aphids, I wonder what you know about the distribution of their plant hosts and whether, presumably, wingedness is beneficial if you can actually get to a new host. So if your host is rare, might you be less winged because that's not going to be a successful strategy?
Yeah, we thought about that a lot when we were doing this biotype screen. And we thought that some of these, we know a little bit about these plants. Some of them are cultivated in huge fields, and some of them are kind of at the outskirts of the fields. And we assumed that aphids that fed on these plants that might be really patchy might disperse more, but that's kind of the opposite of what we found. The two biotypes that we found that had the highest wing induction are also those that are the most widely cultivated. Alfalfa and red clover are really common plants to plant. So that's definitely something we're thinking about, but we haven't quite made a sensible story out of that yet. Yes?
If I understand correctly, in your GWAS study, you use the phenotype you use is the percentage wing
Yes.
in crowded condition, right?
Yes.
Why don't you use the difference
Yes.
in percentage between crowded and solid?
Yeah, we have done that as well. The difference is basically equivalent to the percent winged crowding because they don't produce a lot of winged offspring in the solitary condition. That there are a few genotypes that are kind of weird, 4 or 5 genotypes in our panel that are weird and seem to produce a lot of winged offspring regardless of what we do to them. And we're not sure whether to do a new analysis where we look exclusively at slope or whether to exclude them or how to think about them. Because what we think is important is ultimately how many winged offspring are you producing rather than the slope of the line. If you have a genotype that's producing 85% winged offspring in a solitary condition and 100% in the crowded condition, and you say that the plasticity is 15%, it seems a little weird to call that the same as an aphid that's going from 0% to 15% in our model. But I mean, that's a really good.
In terms of amount of plasticity, it's the same, like 15%, right?
Yeah, in terms of amount of plasticity, it is the same, but in terms of the effect that it has on your offspring, it's not the same. And so
If you have mapped the genes that control the variation of plasticity, then the phenotype should be plasticity.
yeah, and so for almost the overwhelming number of our lines, that is exactly what we did measure. It's the same, and we're working. We're still thinking about what to do with the very small number of lines that have that issue. Yeah, there was somebody in the middle.
Yeah, I was wondering in the wild how common the winged aphids do leave, because if they're leaving the dense population, then fungal infections might not be acting as a strong selection against having wings.
Right. So we measure the immune response in aphids that are not yet adults. And so they're experiencing these high density conditions for 11 days before they grow the wings. The wings are only active when they molt to the adult stage and then can fly away. So that's a good point. But we think they are experiencing these high density conditions and kind of the conspecific disease transmission that happens while they're still juveniles. But I guess a more broader question is how common are these winged aphids in the wild? They're super common and they disperse all the time. And there's some really cool studies from some Chinese groups that basically went with a vacuum cleaner up on a tall building and just sucked in air and counted the number of aphids that they collect. And they collect a lot of aphids, and a lot of them are infected with fungus. So, yeah. Great, thanks. Do you need this?
I do need that. Thank you. All right. Do you want to pull up your presentation? USB, the last presenter.
What's the problem?
The USB connector to the.
It fell, right? So
I can just grab another one so we don't have to search for it right now.
I'm pretty sure I didn't steal it.
That probably just fell. All right, I'm going to go ahead and introduce you while we wait. It's my pleasure to introduce Daniel Schwab. He's currently a fifth-year graduate student in the lab of Dr. Armin Moczek at Indiana University. His research seeks to understand how developmental mechanisms and ecology interact to shape developmental and evolutionary outcomes. He does most of his research on dung beetles in the genus Onthophagus, which is among the most speciose genera in the animal kingdom. When on the lab, Daniel visits local schools and community organizations to present on topics as wide-ranging as the functional morphology of mammal skulls, and insect development and diversity. Today, Daniel will be sharing some of his research on the role of hormones, microbes, and niche construction in dung beetle development and plasticity.
okay. Well, thank you for that nice introduction, and thanks to all of you for inviting me and for coming to see my talk. So today I wanted to begin my talk on phenotypic plasticity with a classic definition of phenotypic plasticity as being the ability of individual genotypes to produce different phenotypes when exposed to different environmental conditions. This is probably the most ubiquitous, widespread, and frequently used definition of plasticity in the literature. It's certainly the one that I grew up with 5 years ago as an undergraduate. But what I want to point out to all of you today is that this definition contains within it certain implicit biases and assumptions about the nature of organisms and the nature of environments. And so, for instance, if we think about organisms as being comprised of multiple hierarchical levels, ranging from the genomic, cellular, the tissue, et cetera, levels all the way up to the organism, and being found within a broader environment, in this definition, we see that we define organisms primarily as genotypes, or think of plasticity as a product of genes and genomes that may be unraveled unidirectionally. In addition, it talks about environments as being something that organisms are exposed to or not exposed to. And so it thinks about environmental responsiveness as being something of a special case, and of environments as being something that's passive, external to, and largely separable from the organisms that experience them. However, I think that recent advances in evo-devo and developmental plasticity literature have suggested that this framework, while really helpful for studying the evolution of plasticity, also has certain limitations. And so, for instance, and I should say this comes from about 2 observations that I'll characterize. The first observation is that environmental responsiveness is not a special case, but can often be seen as a critical feature of normal development. And that plasticity emerges from internally constructive developmental systems that are not simply computed by their genes with a complex G to P map. And so, for instance, if we think about butterflies, polyphenic butterflies in the genus Bicyclus, which they must develop into either one or the other morph, depending on whether they're in the wet or the dry season. So response to environment in this case is a constitutive part of development. It's not optional. At the same time, if these signals from the environment are integrated through the nervous system, which results in hormonal changes, which feed into genetic changes, which then feed back onto higher levels of organization. And so the response of these organisms is highly complex. The second observation that I want to address is that environments are often not as passive, external to, or even separable from organisms as previously considered. And organisms can be thought of not just as internally constructive systems in terms of the physiology and developmental genes involved, but also as externally constructive systems. And so I think a good example of this comes from developmental symbiosis, where we know that organisms may be oftentimes thought of as not just single individuals, but oftentimes as teams or holobionts, where microbiota that are found both within the cells of the organism or external to the organism can send signals to the developing animal or plant that are necessary for the completion of normal development. And so, for instance, a good example of this is the bobtail squid Euprymna, where the development of its immature light organ is reliant upon Vibrio bacteria. And so these bacteria send signals for that organ to complete its development, which in turn sends signals to those bacteria and tells them to bioluminesce. And so, importantly, these interactions are reciprocal, right? The bacteria is sending a signal to the cells of the organism, feed back to the genome, and then feed back to expression of genes in the bacteria. Similar sorts of interactions are characteristic of niche construction and eco-evolutionary feedbacks. And so in these cases, organisms modify their external environment in ways that feed back onto the organism, induce developmental changes that can then feed back onto future bouts of niche construction. Of course, good examples of this, there's many, but here I just highlight the insects, hymenopterans, bees, wasps, and ants with their mounds and their hives. And so within this extended framework, I use horned beetles in the genus Onthophagus to better understand the causes and consequences of phenotypic plasticity. And so today I'm going to address three primary research questions for all of you. The first question is going to relate to those internal constructive processes in developmental plasticity, where I'll be asking what physiological and developmental genetic mechanisms underlie the evolution of nutrition-sensitive horn growth. And specifically, I'll study this within the context of the evolution of allometry and the role of serotonin signaling. In the second aim, on niche construction in the microbiota, I'll talk about how parents and their offspring modify developmental environments and what the consequences of these modifications are for the developmental and fitness outcomes of offspring. And finally, in the third part, I'll just take a brief moment to synthesize some of these studies and to highlight some future directions for both Onthophagus and I think for the field of plasticity in general. And so I'll start here, and I first want to just acknowledge two talented undergraduates in the lab, Justine Christian and Keely Newsome, without whom much of this work wouldn't have been completed. So the first thing that I want to highlight for all of you is that Onthophagus beetles are really an ideal system for studying the evolution of allometry because their allometries vary at multiple levels of organization. So for instance, within a single male, and here I have a standard allometry where we'll have body size on the X and trait size on the Y. And actually, before I show you anything, I want to point out that in our beetles, body size is largely determined by nutrition. There's very little input of genes on overall body size, and so you can readily swap out these two axes. So just think of this as an environmentally responsive trait itself. And so if we look within an individual, we can see that traits vary in terms of their environmental responsiveness on great orders of magnitude. So the Adiagus or the male penis hardly responds to environmental inputs whatsoever. The four tibia, which is part of the legs, are fairly environmentally responsive. And then we have this really complex sigmoidal allometry that's highly environmentally responsive. And this is indicative of a polyphenism. And so we have a threshold here at about 5.01 millimeters. Below this threshold, you develop hornless sneaker males. And above this threshold, you develop horn male morphs. At the same time, these allometries readily evolve, and rapidly so, in nature. And so, for instance, if we look at the threshold at which horns are produced, we see that between Western Australia beetles and Eastern US, the threshold has diverged heritably due to ecological conditions in just less than 100 generations on an order of about 0.3 millimeters, which is fairly substantial. At the same time, if we look at macroevolutionary scales, we can see that allometries have diversified within the Onthophagus lineage in all sorts of directions. So here we have Onthophagus taurus in green. We see that some other species have greater amplitudes or lesser amplitudes, and other species have lost this highly nutritionally responsive horn growth altogether, such as in the case of Onthophagus sagittarius. Now, for the longest time in our lab, one of the things we've been curious about is we've wanted to understand, this is one of the things that I'm most excited about, is understanding how we go from a linear scaling relationship to a more complex sigmoidal allometry. And what we've learned from this is that there are diverse growth regulatory pathways that seem to have been recruited or co-opted into the regulation of horn polyphenism. And so we find that components of the hedgehog signaling pathway seem to suppress the growth of horns in small individuals, whereas components of the somatic sex determination pathway seem to induce disproportionately large horns in large individuals. And further, we know that the insulin signaling pathway can modify the slope of the of this allometry. However, although these pathways have been really helpful in understanding the evolution of sigmoidal allometries, they don't appear to influence the body size threshold at which horns are produced. And so in order to try to identify which factors are underlying these shifts in the threshold, we used some transcriptomic data sets. And we found they implicated serotonin signaling, which is really surprising to us because we know that serotonin is usually limited to the brain. All the serotonergic neurons are in the brain. And we know that horns are developing in epidermal cells that are really, really far away from the brain. Yet, when we look at a microarray, we find that serotonin receptor 1 is upregulated in the horn epidermal tissue of large male pupae in comparison to small male pupae. And through an RNA-seq data set, we find the expression of serotonin receptor 1 and serotonin biosynthetic enzymes differs across populations at key developmental stages. So for instance, if we look at large male pupae and the gene tryptophan hydroxylase, which is the rate-limiting enzyme in the serotonin biosynthesis pathway, we find that Western Australia beetles express this gene at much higher levels than do North Carolina beetles. And this is very interesting because we know these populations have also diverged in where their threshold lies. So in order to see if there's an actual functional effect of serotonin signaling on this allometry, we took a pharmacological approach. And so basically what we do is we rear our beetles inside of 12-well tissue culture plates that are lined with dung. And then we treat that dung with one of three chemicals, either 5-HTP, which increases the amount of serotonin in the system. That's all you guys need to know. AMTP, which should decrease serotonin levels in their systems, or just water as a control. And the first thing I want to tell you just briefly is that we found no effect of increasing serotonin levels on any aspect of the horn allometry. However, when we decrease serotonin signaling using AMTP, we find a shift in the inflection point or the threshold at which beetles produce horns and no other aspect of the allometry. So here we see that the threshold shifts by about 0.2 millimeters from the control. This is especially interesting because this divergence is on the order of the divergences that we see in natural populations. Now, next thing that I was curious to know is whether serotonin is functioning generally as a growth regulator or growth promoter, or if it just plays a role in those horns as those RNA-seq data would suggest. And so I decided to look at two traits that develop at different locations in the body and also have divergent scaling relationships. So we have the elytron here, which covers up the wings. It's produced on the thorax and scales really nicely with body size. We also have the Adiagus, or the male penis, which is located on the abdomen and scales very poorly with body size, as I mentioned before. For both of these traits, we find no effect of either 5-HTP or AMTP treatment on the growth of these traits, suggesting that serotonin signaling has only effects on the production of horns. What I next wanted to do is I wanted to take advantage of the diversification of horns and horn types in the Onthophagus phylogeny. And so as you recall, we have Onthophagus taurus as our focal system, which produces horns on its head and generates horns through a sigmoidal polyphenic form of plasticity. And we know that AMTP can shift this threshold down to lower body sizes. What I next wanted to know is I wanted to look at Onthophagus gazella, which is one of the most ancestral species in the Onthophagus phylogeny. Just like taurus, it produces horns on its head. It develops horns in a sigmoidal polyphenic manner. And so I can use this species to ask whether serotonergic regulation of the horn threshold is an ancestral feature of the Onthophagus phylogeny. I next want to look at Onthophagus nigroventris, which has secondarily lost the production of horns on the head and has novelly produced horns or expresses them through the thorax. And so in this species, we have horns in a novel location, but we maintain that sigmoidal allometry. So I can ask whether serotonergic regulation is co-opted along with the other mechanisms that give rise to this novel horn type. Finally, I want to look at Onthophagus sagittarius, which is one of the most closely related species to Onthophagus taurus. This species produces horns on the head, but it's secondarily lost nutrition-responsive horn growth, this sigmoidal plasticity. And so I can ask if serotonin has an effect on horn growth in the absence of a threshold to set. All right, so first I want to show you the data from gazella. And so here we find that just as in Onthophagus taurus, we can shift the threshold to the left with AMTP treatment. We find the same result for Onthophagus nigroventris. However, for Onthophagus sagittarius, we find no effect whatsoever on the horn allometry. And so in combination, what these data tell us is that the evolution of complex allometries in Onthophagus involves the recruitment of diverse growth regulators from the hedgehog signaling to somatic sex determination, and that serotonin appears to set the threshold at which polyphenic horns, but not linearly growing horns, are induced. And this seems to have arisen early in the Onthophagus phylogeny. And so work in progress. So presently, I'm trying to measure serotonin titers in the hemolymph of different Onthophagus taurus populations. So I mentioned that we have those divergent populations, Western Australia and North Carolina. So I'm trying to get these into the lab now and measure whether the amount of serotonin in their hemolymph differs. And then we're also assessing the effect of serotonin receptor 1 RNAi on the production of horns. I actually have some of these data, but I don't have time to go into them now. So if you're curious, please ask me after the talk. So now I want to move from thinking about the sort of internally constructive physiological developmental processes that allow our beetles to respond to their nutritional environment and think instead about external factors and interactions that these beetles have that can structure their plastic responses. And so for this aim, I want to just acknowledge Haley Riggs, who is now a graduate student at Miami University in Ohio, and Sophia Casasa, who's another graduate student in the MoCheck lab. So I mentioned niche construction very briefly, but I want to give you a quick introduction to the field. So niche construction occurs when organisms systematically modify their environments through their physiology and behaviors. There are lots of good examples of this. The most charismatic ones are probably birds in their nests, flies in their galls, beavers in their dens, and of course humans are wonderful niche constructors. This is thought to be a critical aspect of normal development in diverse species. However, there are few model systems out there where we can readily manipulate their abilities to niche construct or where we know what aspects of their environment they're modifying in specific ways. And we also don't know how these modifications give rise to phenotypic changes or have phenotypic effects. And furthermore, one of the hypotheses about niche construction that has yet to be tested is that developing organisms which are engaging in niche construction are not just doing so plastically, but their modifications may feed back onto the growth of their own plastic traits. And so there may be nice feedbacks between the organism doing the modifying and the organism then itself, its other traits. In addition, we have ecological and population genetic models that suggest that niche construction can heavily influence evolutionary dynamics, so things like the rate of adaptation, the degree to which genetic variants can be maintained or lost in natural populations. And I think these models need a lot of truthing in the empirical literature. But one of the things that I wanted to address with my study system is the fact that we don't really know how the phenotypic effects of niche construction may diversify amongst closely related species or populations. And so I want to begin first by talking about the ways in which adults structure niches for their offspring. And so during the reproductive season, adult dung beetles can basically fly over to dung pads near the air-soil interface. They'll dig a tunnel underneath these dung pads, and at the bottom of each tunnel, they'll build a big ball of dung. And within this, called a brood ball, and within this big ball of dung, they'll excavate what's called a brood chamber. And within that brood chamber, mom will deposit a pedestal made of her own maternal fecal matter. And when the larva hatches, the first thing that it's going to do is it's going to feed on that pedestal before feeding on the rest of the brood chamber. Now we know that this pedestal in the brood chamber contains a complex microbiome. We know it's vertically transmitted from mom to larva via this method. And we know it's distinct from the microbiota that are found in dung, the brood ball, and soil. But what we don't really know about are the functional and developmental importance of these microbes. And so for the longest time, people have hypothesized that microbes might be critical for the development of dung beetle larvae, because we know that dung is a nutritionally recalcitrant resource that's filled with complex polysaccharides like cellulose, very low in essential amino acids. And so there's long been a thought that microbes may be important for their growth. And so essentially what I did was I cultured out the microbes that are in this pedestal. And I'm just going to show you a few data slides to show you the effects of these microbes on the growth and development of our beetles. So the first thing that we find is that when we rear our beetles with microbes, they develop much more quickly than when they are not given microbes. And furthermore, we find that when we give them their microbes from mom, they develop to larger body sizes than when they are not given these microbes. And these are both highly significant effects. And importantly, both of these metrics are highly correlated with fitness, both in invertebrates generally and in Onthophagus beetles specifically. So the time to adulthood is going to determine how much time you have to lay eggs during the reproductive season, how many females you can mate with. And we know that adult body size is highly, highly correlated with the number of eggs that an individual can lay, as well as the degree to which they can provision their offspring. What we next wanted to do was we wanted to try to add a layer of ecological relevance to some of these results. And so we know that beetles can be buried at various levels, at various depths in the soil, ranging from being right at the air-soil interface all the way down to 30 centimeters below ground. If you look at the mean daily temperature range that they experience, you find that near the air-soil interface, they experience massive fluctuations in the temperatures throughout the day, from daytime to nighttime. However, as you go deeper and deeper in the soil, you find that these fluctuations are diminished. And this is important because these fluctuations can be a major stressor. So energetically, it's really hard to maintain homeostasis in the face of temperatures that fluctuate like this. And we know this results in, these stressors result in smaller animals. So what I wanted to do is I wanted to ask, if I rear beetles either with their microbes or without their microbes, and under a constant temperature condition that simulates these depths of about 30 centimeters, or fluctuating temperatures that simulate depths at about 0 centimeters, what is the relevance of the microbes in this case? How do they influence the phenotype? And so what we find is that while there's a significant difference in the time to adulthood when beetles are given their microbes, they develop faster under constant temperatures. Under fluctuating temperatures, there's an interaction effect where the disparity in these responses increases, and those that are not given microbes do disproportionately worse. And so this suggests that the evolution of this of microbe provisioning by mom maybe have been particularly adaptive for populations where they bury their brood balls at very low depths in the soil. The next question that I want to address, however, is whether larvae themselves engage in niche construction. So larval Onthophagus engage in a lot of putative niche constructing behaviors that I've noticed throughout my dissertation. They do things like brood ball repair. If there's a hole in the brood ball, they'll plug it with their dung. They're constantly feeding, defecating, and refeeding on their dung. So they'll spread those maternal microbes that they've inherited throughout their brood ball and then refeed on that dung later on in their development. They redistribute brood ball content, and they actually construct themselves a fairly complex pupation chamber that protects them when they're in this immobile stage. However, the importance of these brood ball modifications and the degree to which their effects may vary among populations and species is unknown. So in order to try to address the phenotypic consequences of niche construction in this system, I took a simple and very novel approach to try to manipulate the niche constructing abilities of our larvae. And so basically we had two rearing conditions. We had a niche constructing condition and a non-niche constructing condition. Now in both conditions, we allowed larvae 24 hours to sample their maternal microbiota because we know that this is important for their development. What we would then do is we would transfer them to those 12-well tissue culture plates that are filled with dung. We would allow them to stay there for 48 hours. However, once those 48 hours elapsed, what we would do is for the non-niche constructing treatment, we would take those larvae out of the brood ball and transfer them to a novel brood ball. And then we would cover this up to replicate the conditions that we see in the brood ball. However, in the niche constructing treatment, we would simply lift the larva out of the brood ball and then place it back in and cover it up. And so we would do this throughout the development of these animals. And so essentially what we're doing is in the niche constructing treatment, we're allowing beetles to modify the same brood ball throughout their development. Whereas in the non-niche constructing treatment, we're giving them a novel, fresh brood ball every 48 hours, which in the absence of niche construction, you might expect to actually be really beneficial to these animals, getting new, fresh dung. One thing that I'll also say is that we know that the age of the dung really doesn't matter in terms of its nutritional benefits that it provides. So the first thing I want to show you all is that if we look at the effect of niche construction on each, we looked at it in 3 different species, Onthophagus taurus, Gazella, and Sagittarius. And in Onthophagus taurus, we find that niche construction leads to a substantially significant effect on adult body size with a difference of about 0.6 millimeters. This is actually a major difference because the range of body sizes that we see in natural populations is actually about 2 millimeters. This is a substantial fitness cost for these animals. For Onthophagus gazella, we also see an effect, though it's diminished relative to Onthophagus taurus. And for Onthophagus sagittarius, we find no effect whatsoever. So we find that there's variation among species in the degree to which they seem to engage in niche construction. What I next wanted to do is I wanted to see if these niche constructing modifications that our beetles are engaging in feed back onto these organisms and the development of their adult traits. And for this, I picked the foretibia because as I showed you before, we know it's responsive to the nutritional environment of the larva. And we also can find it in males and females, so we don't have to throw out one sex entirely. And so what we find, and I should say I have body size here on the X and each species up here, foretibia on the Y. What we find for males of Onthophagus taurus is that throughout the body size range, those beetles that engage in niche construction develop larger foretibia than those that do not. We find a similar effect for Onthophagus gazella, but for Onthophagus sagittarius, we find a reversed effect, which I think is really interesting, and I'm happy to talk about this during question and answers. For females, we find largely similar effects. In Onthophagus taurus, we find that shift in the intercept. We find interesting slopes or interactive effects for both Onthophagus gazella and Onthophagus sagittarius. And so what this suggests is that niche construction can indeed feed back to shape plasticity in the traits of the niche constructor as is suggested in the literature. What we next want to do is we want to take advantage of the fact that the foretibia are sexually dimorphic in this species and see if the degree of sexual dimorphism is modified in the presence and absence of niche construction. So we know in natural populations of Onthophagus taurus and Onthophagus gazella that males have longer foretibia than females. They actually use them to grasp females during mating. And we know in Onthophagus sagittarius, females have just larger, beefier foretibia than the males for reasons that have to do with their ecology. So we want to ask the degree to which these sexual dimorphisms are maintained or modified in the presence and absence of niche construction. For Onthophagus taurus and gazella, we found that under niche constructing treatments, these sexual dimorphisms are maintained, and they're maintained on the order that we tend to see in natural populations. And for Onthophagus sagittarius, we similarly see that females produce larger foretibia than males do throughout the body size range. However, when we looked at the non-niche constructing treatment, what we found is that the sexual dimorphism is completely disappearing in Onthophagus taurus and in Onthophagus sagittarius. And for Onthophagus gazella, the degree of this dimorphism diminishes between niche constructing and non-niche constructing treatments. And so what this suggests is that niche construction affects the degree as well as the presence and absence of sexual dimorphism. And when we know that sexual dimorphisms are responsive to environmental conditions, particularly nutrition, this is the first example that we're aware of where niche construction can actually completely eliminate or even modify a sexual dimorphism. So we're then curious to know whether we can identify a possible mechanism by which beetles are modifying their brood ball environment. So I mentioned that there are a few different things that beetles are doing in their brood ball, all of which are being eliminated when we treat them to the non-niche constructing treatment. And so one possibility is that this nutritionally recalcitrant dung may be conditioned by maternally derived microbiota that our beetles are spreading through the brood ball when they're defecating throughout it. And this is known as an external rumen. And so basically the idea is that by spreading these microbiota throughout their microenvironment, these microbes can digest some of these complex polysaccharides external to the animal. And then when our animals actually feed on those polysaccharides, they're now broken down and are more easy to digest. And there are several examples of this in nature. So in order to do this, we reared brood balls actually under something called larva plus or larva minus treatments. In the larva minus treatment, we allow brood balls to persist for 1 week, either without that beetle or with a beetle that defecates throughout the brood ball. We then sample the brood ball after that 1 week, and then we subject it to something called community-level physiological profiling using biolog ecoplates. So essentially what this is it's a 96-well plate that's cut up into 3 triplicates of 31 different carbon substrates that are commonly found in most soil environments, many of which are found in dung. And so what we can do is we can extract the microbiota, put them in each one of these wells, and then we can measure by the degree of color change the degree to which these substrates are being utilized. And it can give us some sense of the activity in these microbial communities that are being passed down from mom to offspring and then being spread throughout the brood ball. And so briefly, I'll just show you, I'll have all carbohydrates that were significantly utilized here, amino acids and carboxylic acids. And here, these numbers are the degree of substrate utilization. For the larva minus treatment, out of those 31 substrates, we found that only 2 were significantly utilized, galactonic acid and asparagine. However, in the larva plus treatment, we find that community activity increases substantially. And so we find that there are 10 substrates that are significantly utilized, and those that overlap with the larva minus treatment are significantly utilized. And what's especially interesting about this is that these molecules are the disaccharide components that make up cellulose, chitin, and pectin. So though we still need to do further experiments to see if these microbes are actually able to digest these molecules, these data are very suggestive. So in summary, we now know that for Onthophagus larvae, niche construction generates environmental feedbacks that are a normal component of development, are variable among species, and seem to feed back onto body size as well as onto scaling relationships. It may be reliant on developmental symbionts, though other mechanisms are possible. For instance, I mentioned that these beetles construct a pupation chamber. And so for most systems, we really have no idea about the functional consequences of these pupation chambers, and that's something that I still want to test. So now I just want to spend the couple of minutes that I have left trying to synthesize these different approaches to studying plasticity in our beetles, and also suggest some future directions. So historically, the way the plasticity researchers have studied internally constructive processes, so things like nervous system signaling, endocrine signaling, the differential gene expression that give rise to plastic organismal phenotypes, have done so independently of a subset of researchers that study things like niche construction, the influence of learning on development and social interactions, as well as symbionts. And these pursuits have been really productive. Like, our lab has done a great job of unraveling the developmental genetic and physiological underpinnings of polyphenism, right? But what I think is the case is that the lessons that we're learning from Onthophagus, as well as many other systems that I don't have time to go into today, suggest that a more integrative and holistic account may be worth exploring. I think particularly in our system, the writing is on the walls here and suggests that niche construction can influence morphogenetic processes and that symbiont-derived environmental modifications can influence things like endocrine signaling. It's hard to see how they might not. And in addition, we think that there may be some feedbacks onto social interactions that are coming from things like serotonin signaling. So the way that I would argue that we need to start thinking about organisms is not approaching any one system solely from an internally constructive perspective or an externally constructive perspective, but to start to study organisms or more regularly study them as sort of a continuum amongst these different processes in order to understand the generation of organismal phenotype. And so I think this account is particularly important because I think it has the potential to highlight interactions whose explanatory value really remains to be fully recognized. And this will be particularly important for identifying the developmental origins of complex plastic phenotypes, and also for identifying their evolutionary consequences and the ways in which they can bias the outcomes of developmental evolution. And so what we're hoping to do in the near future with our system is we want to start rearing larvae with either their microbiota and niche construction allowed or suppressed to generate developmental transcriptomes and metabolomes and identify candidate genes and pathways. With that, it looks like I'm out of time. So I just want to thank my lab, including my advisory committee and my PhD advisor, Dr. Armin Mochek, my research assistants, my many sources of funding. And with that, I'll take questions if I have time. Thank you. And here's a picture of a beetle brain. Do I have time? Okay.
Was there any evidence with your serotonin treatments that behavior changed?
So it depends on the amount of serotonin that you give them. So if you give larvae a ton of serotonin, they'll really slow down in terms of their eating. But we were able to modify the amount of serotonin or manipulation such that we didn't see these phenotypes. So there doesn't seem to be any obvious negative consequences of manipulating serotonin.
So like in the adult stage, you didn't see.
Oh, I see what you're saying.
Yeah.
No, so these effects don't carry over, and I think this is because most of our these pharmaceutical chemicals are probably used up across pubertal therapy. There's no reason to think they might have carried over into the adult stage. We didn't even look at adult phenotypes.
You might have to take your new dick off because it's been rubbing against the microphone. Stop it. Drama. Sorry. Okay. Okay. Sorry, I guess you can. That's perfect.
All right, it's my pleasure to introduce our last speaker before the coffee break, Clara Richoff, who's currently an assistant professor at the University of Kentucky. Her research integrates across behavioral ecology, genomics, and neuroscience to understand the evolutionary consequences and mechanistic basis for behavioral plasticity. In the past, she's done research on reproductive strategies in the golden orb spider, and her lab currently focuses on the honeybee. When not in the lab, Claire spends time sharing her research with local beekeepers, and she also helps advise a local Lego robotics team. Today, she's going to be sharing some of her work on aggression in the honeybee. Thank you.
Thanks. Thanks for that introduction, and this has been a fantastic day of talks. I'm sort of overwhelmed and a little bit tired at the point that I have to give my talk, which is unfortunate. But anyway, I'm hoping this will kind of touch on a few points that others have made throughout the day. I've certainly picked up on a lot of themes. And this will be a little bit light on the sort of eco-evo side. It's a little mechanism heavy, but I'm hoping that will sort of feed back and aid the development of some new theory. Okay, so the first panel discussion really set up this question very nicely, which is this recognition that behaviors are really special phenotypes from the perspective of phenotypic plasticity. And I would argue there are a lot of reasons for that, but one is that they show an exceptional degree of plasticity. So for example, within a single individual's lifespan, they can turn on and turn off behaviors associated with reproduction, like territoriality or maternal care behaviors. And then even on a much shorter time scale, there are behaviors that are more activational in nature. So they can be activated and terminated in the time scale of minutes or even seconds, like this peacock spider mating display or this anti-predator display by these mantids. And certainly, it's pretty easy to argue that when we compare behavior to some other phenotypes that we typically consider in the context of phenotypic plasticity, they're much more variable. Most of the other phenotypes are morphological in nature. Okay, so I want to highlight what I see as some sort of key features of behavioral plasticity that make it a little bit exceptional. And the first feature is that environmental influences on behavior span a lot of different time points in an organism's lifespan and lifetime. So for example, you might have environmental effects that influence the mother while an offspring is developing. That offspring will then be further influenced by developmental environment and the quality of maternal care. And then finally, behaviors are really special because they continue to be environmentally responsive throughout adulthood. So one thing to keep in mind, because of these multiple time points for environmental influence, is that these environmental influences are actually going to be non-independent and have intersecting timescales of effect. So I'll explain what I mean by that with a sort of schematic. So imagine you have a phenotype. The baseline level of that phenotype might be set by a maternal effect, so that might differentiate the orange and blue lines. But then you can have additional effects that happen during development that further modulate that phenotype. And there's plenty of evidence that both of those types of environmental inputs can last throughout the lifespan of an individual. But then we also have effects that are going to happen during adulthood, so additional environmental inputs that will further modulate those phenotypes on the timescales of days, minutes, or seconds. And one reason that these environmental influences are non-independent is because they tend to act on similar underlying mechanisms. And I think Laura's talk gave a cool example of this, looking at some of the transgenerational effects of a predator threat, comparing those to sort of personal experience of the predator threat, and showing that those modulate some of the same genes. So given that we know that some of these environmental inputs are acting on the same mechanisms, what's actually modulating these temporal dynamics? So why is it that some environmental inputs have very transient effects on behavior and others have much longer lasting effects on behavior? I think there are implications for this question on a variety of different scales. So for example, even with human health questions, one standard issue is why can we remediate some environmental effects and others seem to be sort of more stable. But also when we're thinking about phenotypic plasticity theory, these sort of mechanistic interactions may be limitations to variation in phenotype over time. So there are a few different types of explanations for how time is actually encoded in the organism, leading to different timescales of effect. So one is that these environmental inputs might be acting at different levels of organization. And Daniel used levels of organization to sort of talk about population and species level differences, but here I'm talking about processes within a single organism. So one really good example of these different levels of organization comes from the brain. So this is the electrical versus molecular scale of organization. So you're probably all familiar with an action potential. So when an organism perceives an environmental stimulus and they execute a very rapid behavioral change, that's electrically mediated. So the action potential is a signal that occurs on the timescale of milliseconds. But when you receive an electrical impulse to a neuron, it also induces a change in gene expression in that same neuron. And this is sometimes referred to as a genomic action potential. So this is a change in RNA abundance over time. And that's usually a longer time scale, sometimes hours, sometimes minutes. But the idea here is that for a single neural circuit that might modulate a behavioral phenotype, rapid changes in behavior are electrically mediated and longer term ones are mediated by the molecular state of those neurons. So you may be familiar with some other examples of this type of timescale difference. So for example, epigenetic mechanisms, changes to the chromatin, have been proposed as sort of more stable mechanisms that might modulate gene expression changes at longer timescales. Brain structural changes are another common example. This underlies sort of this idea of why there's a developmental critical period. So environmental inputs during brain development may have long-term effects on function. It's notable that these ideas are all out there to explain timescale variation, but there's very little evidence that really ties these particular mechanisms to predictable levels of timescale variation. So there's a lot of work to be done in that area. And the other piece of this that I think is really missing is an understanding of how these levels of organization actually interact to modulate behavior over time. So to use the electrical molecular example, an electrical signal causes a change in gene expression, but presumably that's also going to modulate properties of the neuron longer term. So it's going to affect protein concentration in those cells, and that should feed back and affect the signaling potential of those neurons. So that sort of closing that feedback loop is another area where we need some more research. Okay, so I'm going to try to approach some of those issues in my talk today in the context of honeybee aggression. You're probably all familiar with phenotypic plasticity in the honeybee, particularly developmental plasticity of females, where you can give rise to either a queen or worker phenotype depending on nutrition. But the other cool thing about honeybees is that workers are actually highly behaviorally plastic throughout their lifespan. And this is necessary for almost every colony function. And honeybee aggression is a particularly cool example of this because defending a colony against a mammalian predator, probably one that's a little more effective than Winnie the Pooh, requires hundreds or thousands of individuals to coordinate their activity. So this is a highly socially regulated behavioral phenotype. And I won't go into all of the contexts for social regulation, I want to focus on two that show very clear differences in timescale. So the first is an acute context for variation and aggression, and this is mediated by a specialized alarm pheromone. So the way this works is there's a specialized caste of bees called guard bees that monitor the colony entrance for threats. And when these bees sense a threat, they will alert their nestmates using a chemical cue, an alarm pheromone. So this cue is perceived, if they actually waft it into the colony, it's perceived by a subset of bees called soldier bees. That's also another specialized caste. And those bees will induce a rapid aggressive response. So this is a context of social modulation of aggression that occurs very quickly. But there's another longer-term timescale for social modulation of aggression that has to do with this really interesting behavioral maturation phenomenon that we see in honeybees. So when bees first emerge as adults, they tend to perform tasks inside the hive, like nursing behaviors. And then as they age, they transition to performing tasks outside the hive, like foraging is an example of that. So what's interesting about this is an individual bee's movement along this trajectory is dependent on social information and the needs of the colony. So they can accelerate their maturational process, they can slow it down or even reverse it, depending on what the colony needs. They can also skip over certain tasks if those aren't required at the time for the colony. So we can use this as a context for social modulation of aggression at a longer time scale. Here, the differences in behavior are much more stable than what we see in the acute time frame. So essentially what we see is behaviorally older bees on this end of the spectrum tend to be more aggressive and more responsive to alarm pheromone in general than the younger bees. So that's our long-term context for variation and aggression. Okay, so if we want to start to get at some of the mechanisms that may distinguish these two different timescales for variation and behavior, we need to talk about a candidate mechanism. And I want to tell you about one I've been working on for the last 5 or 6 years, which is this relationship between aggression and brain energy metabolic state. So we don't think a lot about brain energy metabolism in the context of behavior, but it's important to note that the energy state of the brain modulates signaling, much like changes in neurotransmitter concentration or receptor numbers. And what we find in the context of aggression is that bees that are in a high aggression state show a very distinct metabolic phenotype that's known as aerobic glycolysis. And I'm not going to go into detail about this phenotype. What I want you to know for the sake of this talk is that we see this pattern of decreased oxidative phosphorylation in the high aggression brain. And hopefully that pathway rings a bell for those of us that took biology and biochemistry. So this is a major ATP producing pathway that takes place in the mitochondria. So everyone's a favorite acquired organelle. I guess that's what Holly would think. But so basically what we have is this pathway is showing decreased activity in the high aggression brain. And notably, OXPHOS is really important for producing ATP in neurons specifically. So we have a lot of evidence to implicate this phenotype with aggression. The bulk of that evidence is transcriptomic. So when we look at brain gene expression patterns comparing bees exposed to alarm pheromone versus an unexposed control, or older versus younger bees, what we see is a decrease in expression of genes encoding OXPHOS proteins. We have some other lines of evidence. This is from a metabolomics study where we measured metabolites in the brain of bees exposed to alarm pheromones. This is 5 minutes and 60 minutes after exposure. This is just a heat map of metabolites, and what you can see is there's a shift towards aerobic glycolysis that seems to begin really early at a 5-minute time point and then becomes even more severe at the 60-minute time point. And then finally, on the sort of short-term time scale, we've also shown that if we pharmacologically inhibit oxidative phosphorylation, we can cause an increase in aggression. So for the sake of this talk, I think one of the interesting features here about this mechanism is you'll notice, at least on the transcriptomic level, that this mechanism is very consistent across timescales. So regardless of short or long-term differences in aggression, we see that we have the same phenomenon of decreased oxidative phosphorylation, at least transcriptomically. So what is it that actually differentiates these timescales? And this is sort of the key question that I want to talk about today. Well, there's a long distance in terms of biological organization between RNA and behavior. So one way to try to start to get at the mechanisms that differentiate timescale is to start thinking about how metabolic shifts actually impact neural signaling. And for, in the case of oxidative phosphorylation, we chose to focus on mitochondrial function as one of these intermediate levels of organization. Mitochondrial function is interesting for a few reasons. So mitochondria house oxidative phosphorylation, but they do other really important things in neurons. So they're involved in calcium buffering, maintaining redox balance in the neurons, and their function can change really rapidly depending on the needs of neurons and signaling events. So this seems to be a potential level of organization that could be integral to signaling and gets us closer to understanding the functional outcome of a shift of metabolism in the brain. Okay. So for the sake of this study, what we wanted to do was extract and analyze intact mitochondria for their functional correlates with aggression. And to do this, we used a fairly, well, expensive and somewhat sophisticated machine called a seahorse bioanalyzer. And this machine is nice because it allows us to measure oxygen consumption rate. So remember, OXPHOS is an oxygen-consuming pathway. So we can measure that in intact mitochondria in a high-throughput way. And the other cool thing that I'll come back to about this particular machine is that we can also manipulate energetic precursors available to mitochondria in vitro and look at effects on mitochondrial function. And this is work that I've been doing over the past year with Pat Sullivan, who's a researcher in the medical school at University of Kentucky, and his postdoc Amendra Bakari have been helping with this. And Pat complained about the picture I had of him, so he gets to be Batman until he gives me another picture. Okay, so the first thing we did was look at the correlation between aggression and mitochondrial function in the brain. And we designed two pretty simple experiments to do this. So the first was a context for long-term variation in aggression. Hopefully you guys have memorized this by now. So we have returning foragers as our older group. We also pull out emerging bees from the colony and collect 2-day-old bees to be our young treatment for this long-term variation in behavior. For the acute context, we used the alarm pheromone exposure paradigm. So we collected control bees from the colony that are undisturbed, and then we expose the colony to alarm pheromone and collect some of these responding bees 5 minutes and 60 minutes following that exposure. So for this type of analysis, what we have to do is collect these bees and extract, we dissect their brains very quickly, fresh, and then extract their mitochondria and stabilize the mitochondria in a buffer so that we can measure acute changes in function. Okay, so the first result we got, and this was the first data that I had in my own research lab, gave us the exact opposite results than we had predicted. So this was actually pretty surprising and interesting. So here, this figure just shows respiration, oxygen consumption rate measured with the seahorse bioanalyzer. And when we look at older bees, we actually see a significantly higher rate of respiration compared to young bees when we look at intact mitochondria. So this was the opposite of all the rest of the data that we have relating aggression to brain metabolic state. But we repeated it a few times, and it seemed to be a fairly consistent result. However, when we look at the acute timescale, we actually see a pattern that's different and one that we're kind of more familiar with. So when we compare the control bees to bees exposed to alarm pheromone, we see a rapid decline in mitochondrial respiration. And then you can see that decline persists at the 60-minute time point with some variation, which might suggest that some bees are mitochondria are sort of coming up back to the normal level of respiration. So on this simple sort of mitochondrial scale, it seems that the mitochondria function might actually differentiate these timescales for plasticity and behavior. And the other thing that you'll notice is that we have evidence that the physiological and transcriptomic levels are diverging. So they seem to be allied and match one another in the case of the acute alarm pheromone exposure. When we compare old and young bees, we see this divergence. Okay, so we have this correlative relationship between mitochondrial function and aggression that we've measured with the seahorse. But we also wanted to try to do some causal manipulations of mitochondrial function that were sort of fairly naturalistic and non-invasive. So we actually chose to use dietary manipulations for this. And dietary manipulations may sound a little indirect for manipulating a specific physiological pathway in the organism. But there's really a growing body of research in neuroscience that's examining the role of diet on mitochondrial function in the brain and is showing direct implications for cognition. And for a long time, people have known that diet can be used to treat diseases associated with neural signaling, for example, epilepsy. So a ketogenic diet, or a diet that's high in fat and protein and low in carbohydrates, tends to enhance mitochondrial function and can be used as a treatment for epilepsy. So in general, that's the pattern that we see. If organisms are fed diets that switch them from metabolizing sugar to fat, then it actually can enhance mitochondrial function in the brain specifically. And I've noted here that essentially what you're doing is providing mitochondria with non-carbohydrate energetic precursors. An example of those are ketone bodies, which is a product of fat metabolism. So we set out to see if we could manipulate brain mitochondrial function in honeybees using diet. And the first thing we needed to do was design a ketogenic honeybee diet, which wasn't trivial. They don't eat dung, at least, but they are vegetarians. And so their primary protein source is pollen. But pollen has a mix of nutrients, including a lot of carbohydrates, which bees really love. So it's kind of difficult to control that diet. The other thing they eat, of course, is honey, which is almost all sugar. But it also has pollen and trace secondary compounds from plants, other things that have been shown to influence insulin signaling and metabolism. So I took a very simple approach here, which was just to perform a diet restriction treatment. So here we're feeding bees high and low carbohydrate or low sucrose diets. And diet restriction is a typical form of a ketogenic diet that can actually affect mitochondrial metabolism very rapidly. So we did a 6-day diet treatment here, and then we measured effects on aggression. And we found a very robust effect of diet. So bees fed this low carbohydrate diet show a significant increase in aggression relative to the high carb diet. And what's interesting about this, we actually did a second experiment. I should mention, this pattern resembles what we see comparing old and young bees, where older bees are more aggressive and they show increased mitochondrial function. We did an additional experiment where we actually switched the diets in the last 24 hours to look at how acute diet restriction might affect aggression. And we found that that had no effect on behavior. And this was good because it suggests we're not seeing a starvation effect here. But it's also a little surprising because in other species, namely mammals, ketogenic diets have a very rapid effect on brain mitochondrial function. So to probe this a little bit further, we decided to take an in vitro approach by providing mitochondria in vitro with a ketone body, beta-hydroxybutyrate. So instead of asking the animal to metabolize its own fats into ketone bodies, we cut out this middle step and just provided them the ketone body directly. And when we did that, this is what we found. So this is respiration again, oxygen consumption rate for bees exposed to, what's going on with the laser pointer? Bees exposed to BHB, or their mitochondria rather, for old and young bees. And we see that older bees show a significantly enhanced mitochondrial function when treated with this ketone body. Young bees, interestingly, show no effect at all. So it looks like bees' mitochondria in their brain respond very similarly to other mammals, including humans, where exposure to these ketone bodies actually enhances respiration. But surprisingly, this is age-specific in the honeybee, where older bees show this enhanced mitochondrial plasticity. And I'll come back to that, but it could be that that enhanced plasticity is playing an important role in modulating behavior. I wanted to mention, we also went back to the field and fed these ketone bodies directly to bees and found that within 24 hours, that also enhances aggression. So we kind of closed the loop on that hypothesis. So to summarize, in terms of long-term effects on aggression, more aggressive older bees show enhanced mitochondrial function, and the physiological level appears opposite of the transcriptomic level. Then when we look at long-term and acute shifts in aggression, they seem to diverge at mitochondrial function. So transcriptionally, they look very similar, but at the physiological level, they're different. And moreover, we have a mismatch between the physiological and transcriptomic level for older bees versus younger but not alarm pheromone exposed bees. And finally, this interesting phenomenon where older bees seem to show a higher degree of metabolic plasticity in their neural mitochondria. And that's a result of these BHB treatments. Okay, so given all of that pretty dense information. How can we return to this idea of how metabolism is linked to signaling and interpret this variation in metabolism at these different levels of organization? Well, I mentioned that mitochondria do a lot of things in the neuron other than produce energy. And one really important role they play is in calcium buffering. So when there's a signaling event in neurons, it induces voltage-gated calcium channels to open. This influx of calcium is really vital for vesicle docking and neurotransmitter release into the synapse. There are a lot of secondary messenger cascades that are calcium-induced. However, calcium can also cause excitotoxicity in the neuron, and unregulated calcium content can actually impede signaling. So one of the really important things mitochondria do is they take up and sequester calcium, and they can release it back into the cytosol. So that's a way that they can regulate signaling. And what's interesting about the relationship here with mitochondria is that increased respiration actually enhances this buffering capacity. So when we're thinking about old versus young bee mitochondria, this little green blob is a symbol of oxidative phosphorylation. And without getting into too much detail, if you recall, what this pathway does is actually pumps hydrogen ions out of the mitochondrial matrix. Those can flow back in to generate ATP. But one thing that happens when those protons are pumped out is it creates a negative electrochemical gradient. And that facilitates an influx of calcium for buffering. In the young bee mitochondria, of course, where we have lower levels of oxidative phosphorylation happening, this buffering capacity is much lower. So one way to think about this is that younger bee neurons may have sort of a noisier system when we think about calcium levels, and that may actually inhibit signaling related to aggression. And this hypothesis could also apply to the short-term change that we see in mitochondrial function, because there's evidence that rapid calcium influx, such as what you would see during a signaling event, can cause a temporary decline in mitochondrial function. So what I think is happening here is sort of an interplay between the metabolic level and the sort of mitochondrial functional level. And there are potentially different implications at these different scales. Okay, so I wanted to just finally return to this question of interaction of these different levels of organization and modulating behavioral phenotypes over time. And so what we have here is a model where we get a signaling event that causes an acute decrease in oxidative phosphorylation, but it also causes a change in expression of genes that encode OXPHOS proteins. And presumably, at some point in time, that change is going to feed back on mitochondria and influence signaling. We don't really know what that feedback does. It could be that the new transcription resets the metabolic state of this neuron, or it could be that it changes the metabolic state following this acute experience of an aggressive interaction. And there's evidence in the honeybee, for example, that a colony that's exposed to a predator remains aggressive for a period of time following that experience. So we don't know much about the time course of this feedback and how these two levels are regulated, but we could address that with a pretty simple experiment, just looking at the exposure to alarm pheromone and how that affects mitochondrial function and transcription over time. The other thing that's, I think, intriguing here is that we have this relationship, a positive relationship between OXPHOS and transcription for the acute time scale, but that might be reversed actually over the longer time span comparing older and younger bees. So that could suggest that there's a different regulatory relationship between these two levels, but there could be other things going on as well. So for example, I've kind of suggested throughout this talk that there might be some sort of continuous relationship between the mechanisms that regulate short-term and long-term changes in behavior. Like somehow, if you expose an animal enough to some sort of short-term stimulus, that the brain of that organism may actually shift towards looking like an older bee brain. So that's one possibility. But it could be that there are very distinct differences between the brains at these sort of ends of the spectrum associated with stable changes in behavior. So for example, older bees show structural changes in the brain. They may also show differences in sort of the metabolic requirements. So it could be that there is sort of a disconnected relationship. And in fact, these regulatory mechanisms do change quite a bit comparing the acute and long-term timescale. Yeah, okay, so that's pretty much what I wanted to talk about. I wanted to make sure to acknowledge Joseph Palmer, my technician, standing in front of all these freshly built bee boxes. He helped me go from nothing to data in like 5 months, so I appreciated that. And I also want to acknowledge Patrick and Hemendra for their help with the work. And Jean Robinson and Christina Grosinger are my postdoc advisors. That's where I started a lot of this work. So I'll take questions with that.
So do you think the aggressive behavior in the OB is a byproduct of aging or is actually adaptive?
Is it adaptive?
Is adaptive or just a byproduct of aging? I mean,
yeah, that's an interesting question. I think that, I mean, aggression is certainly adaptive in that it's highly organized and ecologically responsive. I think it would be hard to argue it's not adaptive. As to why those are the bees that perform those behaviors, that could certainly be correlated with age. You know, so the metabolic demands of foraging and the metabolic demands of responding to an aggressive stimulus are likely very similar. So I would say on a colony level, it's adaptive. On an individual level, it probably is correlated to pleiotropy with other traits.
So, thanks. The Western European honeybees are more docile, right, than Africanized bees. Is there any evidence that that is the result of changes in diet as the bees migrated out of Africa?
That's a good question. So there is evidence that the mechanism is probably, the brain mechanism could be similar. Yeah, I didn't really talk about that. One of the sort of dream extensions of this work is to look at how landscape nutritional levels may feed back to affect behavior. So that is related to that question, I think. I'm not sure. Yeah, I think one of the things we don't know much about is how like diet variation affects behavior. So diets differ not only in the sort of protein and carbohydrate ratios, but also plant secondary compounds seem to play a really critical role in modulating gene expression. So any of those could be a possibility. Yeah, that's a cool question.
Sorry.
Okay.
So first off, thank you. Very interesting. You're looking at all of this in honeybees, the most eusocial of eusocial bees. I'm curious if you could speculate on how some of these things might change as you move to something like bumblebees, which are eusocial but not as quite as advanced, or even when you get something like solitary bees, like what some of the signaling cues might be if they're not getting them from, nest mates. Because certain bees really do live alone, and certain bees live communally. I'm certainly aware that you have data for honeybees. I'm just kind of curious what your thoughts might be on what happens as you go across that gradient of sociality.
Yeah, so that's a great question for a lot of reasons. So one thing, we do have evidence that this mechanism seems to be conserved in Drosophila. So the relationship between oxos and aggression seems to be conserved. We also have correlative evidence in fish and mice that it's conserved. So the brain mechanism could be very similar, irrespective of sociality. In terms of the cues, that's also a cool question. I will say that we can induce aggression a lot of different ways, socially and asocially. So we can pretend to attack a colony, or we can expose them to a social cue, or a combination of the two. And we seem to get consistent sort of phenotypic results at both the metabolic and behavioral level doing those things. So I think that this could be a very generalized mechanism for modulating aggression. And one thing that you may have noticed is I'm talking about aggression on the whole brain level here. So we're not looking at individual neural circuits. It's also possible that this metabolic plasticity is a way to modulate behaviors in general, but maybe in specific circuits that govern particular behaviors and not others. So there are a lot of different layers that could generalize that mechanism.
But before we release you all to go drink coffee and eat snacks, we have one more person we really need to thank. Carol, will you come in here?
up here with me or not, but today has run incredibly smoothly. Everything like clockwork. Like, we got to do the really fun part of this, but Carol is the person who actually made everything here possible and happen. So the committee is so grateful for all your effort and work. We have a tiny little present and a card.